Okay, good afternoon. Just in case somebody don't know who I am, I'm DeWei Li. I have to speak up. I work at the Valley Lab in Windsor, now here. And I joined station from 2004. Right now it's close to 18 years. Time flies, I don't realize, so many years pass already. I got my bachelor degree in China, in Nanjing, and master from Lehigh in Canada, and PhD from University of Waterloo. But I worked in industry for a few years, got a different experience, and eventually end up here. I will explain how I got here. Then today I will talk about some new fungal species and new diseases. The reason is, when we find some new fungal species, they are not only new for the mycology aspect, but from plant pathology aspect, they cause some problem. Doesn't work. I have to use this. Okay. The first species I want to talk about is the formerly we call Striatibotrys. Then this one is the Striatibotrys is a very interesting fungus. Before we talk about that, we'll talk about about fungi and the biodiversity. In 1990, then it was estimated there are 1.5 million fungi in the world. And the last 2 years ago, then they redo the stuff, then increase the number to 2.2 to 3.8. Some other studies show even close to 5 million, but I will say this number. And the reason is before the estimate is based on the plant, not the soil, not aquatic ecological habitat, marine situation. But last couple of years, many new species described from aquatic condition or environment and marine. It's incredible. Many of those fungi are hyphomycete. The Striatibotrys first described by, I even cannot see this person, Koch. Then you can see it's 1837. Then, but this fellow, is very interesting, very productive mycologist. And but unfortunately, he died. When he collect specimen in North America, especially United States, when he returned, the ship sunk, and he died with all the members and all his specimen. Then, but he left some specimen. I borrowed the type specimen he described for this genus, and but I only found 3 Canadian. It's useless. It's too bad for so many years. Another one is a similar group, Mamnoniella, described in 1923. Those 2 genera in 50 years, then people always argue whether they should belong to the same genus or 2 separate. Until 2000, then Vesper and Hawkland use ITS to show then they should combine together.
The screen is moving on Zoom for some reason.
Oh,
so let's try this. Stop the share. Stop the share. Okay, now I need.
Can you hear me at the back? My voice? Wow. Okay.
See that. Okay, let's try that.
No, basically. Won't move.
Did it change? All right. Sorry.
During 2000, they show those two should be merged together as a single genus. But the key part is in 1999, if you remember, there's a lady from Texas sued the insurance company because Striatibotrys chartarum was found in her mansion. And the house is a replica of the house in the movie Gone with Wind. They consider is 12,000 square feet. It's a huge one. That's a big lawsuit. And for that house, then they call black mold. And 2000, then all the major media cover this event. It's incredible. That's the reason why I came to United States, work for the company. They really need mycology to identify some indoor mold. Then I tried, then and eventually when I finished, I think I'm not the person who work for industry. Then I prefer research or teaching. But anyway, then during that period of time, then another thing is Dearborn, the medical doctor and the professor, then he studied this problem and associated with Striatibotrys chartarum. Then, and there are some cases he studied. But later on, then the EPA withdraw the support for his conclusion. That's quite bad, actually. But people still, then because the mycotoxin produced by Striatibotrys and people that continue work on the medical aspect, then there are some new development. But that's beyond the topic of this today's talk. Then Striatibotrys, then right now it's a new family for that. Before 2016, then actually I'm the one of the author for this paper, then only consider is one genus. Then we studied 76 different species, published this paper, then in fungal diversity. Then, but a Dutch group, they use this, then separate into many different genera, then which make morphology really doesn't work. If you want to see find a Striatibotrys, that's fine. If you want to see find, for example, this thing will be very hard because the morphology doesn't work. Then in this new system, this the name they publish. They still have the Striatibotrys, but you can see there's although they use the taxonomy, but there are quite a few different genera. And the tricky thing is that they sequence many different loci. They sequence this one also, but they only use for the first analysis. The rest, they didn't use this. Then we use this in this paper. We consider they should belong to same genus. But I'm wondering why they skip this, for other analysis. And that's why I have all the data. When I have time, I want to rerun the analysis to see whether I can get the same conclusion. Then I will see why this one is, although it's a new species before, we don't know which one they belong to. Then the story go back to 2007, when I studied the Striatibotrys, then I ordered two isolates. Then one is from Canada, one from Cornell. Then, and by then, ITS was done. Then we show it belonged to neoeucylindrosporus, that species. Then by then, but I noticed there are some morphological differences compared the type specimen. But by then, only one locus, the information, I cannot draw the conclusion, but it's still in my mind. But after 15 years, I think, well, maybe I should take another look. Then this is why from morphology, we study those fungal structure And later on, I will show you what those mean, and it's very hard, the jargon is incredible. But we use this to take some photos, then, and we did the 6 loci. Thank Neil, and he's a major part of this, and did the alignment and but connect everything together. But we have to align the individually and trim them and to do the analysis. The analysis for any new species, we have to use 2 or 3 different methods to get the same conclusion. If we don't do that, then it's not solid. This is why we have to use 2 different methods then to do the analysis to draw the conclusion, to verify it's truly a new species. Then this one we choose as out group to compare with. This is the tree. Okay, this is the 2 athletes here. You can see they are separate from the 2 closely related. This one, actually, this one is described by myself also. There are some story, interesting story. This is the one is described by the Dutch group. And but those 3 share some similarity, but there are morphological difference. The molecular aspect, you can see they are supported by the analysis, then 2 different methods. Okay, this is the name we proposed, and the authority. Then we include the 4 authors. You can see this is a collaboration. Okay, not only myself, then Neil, Bob, and this is Cuban mycology and myself. Then, but if we want to publish a valid one, we have to include this number. Then this is collected by Cassie Hodge. Then the people from Cornell may know, probably you know her, is a Canadian lady. I guess when she was doing PhD, collect this specimen from mushroom, rotten mushroom. Then another one is collecting Alberta. Alberta is the place I taught plant pathology for 5 years there, and from Aspen. But the 2 athletes, eventually we found the morphological differences and the phylogenetic difference. Then you don't have to read a lot of jargon, but just to focus on the one I highlight, those show the differences with the close related one. This one is a long cyneniophore. Cyneniophore is the one produce cynenium, on which cynenium were developed. Phyllid is the one is the mother cell to produce the cynenium. This one is very interesting. Then they do show the extension from the phyllid to develop another cyneniophore. The cynenia in different shape. Some is this, but they do have this very interesting shape, the dumbbell shaped one. And this is the site. Sexual stage unknown. We only find the asexual stage. This is the fungus. You see the cyneniophore, this one up to here, and this part is a phyllid. Those are bunch of cynenia. They are slimy. Okay, it's very hard to become airborne. Then you really need to, wait until it dry up, then become airborne. And you can see the different shape of the spores. The key part is the striation. Are you able to see the striation on the surface? Only this one doesn't have it. There are some is clear, but if but this part is a very interesting one. In the whole genus or in broad sense, this is the only one I observe. They can extend from phyllid, develop another cyneniophore. Okay. Okay, this is the one I described the year before. Then, and they call this name. This is the specimen collected by George Barron, well-known mycologist from University of Guelph. Then, but by then he called cylindrospora. But however, I borrowed the specimen, type specimen of cylindrospora. I compared, it's different, totally different. This is why I name a new species. And right now, you know, they moved to, based on the Dutch group, they moved to this genus. Interesting thing is, when I searched the literature to describe new species, somebody described it in the thesis, 1954. But it is not a valid description because the thesis is not formally published, won't count. Then I tracked this person down. By then he was mid-80, retired for several years. Then I called him, you know, you have this new species published in your dissertation, whether you want to republish it. Then we can work together. He said, no, go ahead. You know, I retired a long time ago. I'm not interested in this. Then he just want to enjoy his, you know, retirement life. And this is why. But anyway, I include all his information in the paper and to give him credit. He's the first one found this one. Okay, this is this species. And close related one, oh, can you see this is really cylindrical and with the striation and much bigger, much longer, okay? Another close related species, this one, the difference you see, very short cuneiform, okay? And the fat, much wider phyla. The shape of the spore is similar, but is this one even shorter than the one I described. Then this is why the three species can be differentiated. Boy, I have to speed up. Next one. This one actually not much story to talk about it. You know, very straightforward. This is the type specimen for this genus. Then based on the name, you can see what is the major part? The shape of the arrangement of the spore and the theta. If you read this part, then you know that. Then yes, this is described, you know, then proposed in 1885. The type species is this one. But however, this one can be linked to the sexual stage. And so far, there are some species described, and but this one is very interesting. This is indoor fungus. You can see where this collected. Then it's from ceiling fan and in Boston. But you know, this is the one sent to me by one of my former colleagues because they cannot determine its name, which name or even genus can be placed to. Then this is why we did some work and to describe it. But for this one is we use MEA and VA. You were wondering why we use two different medium. Most of mycologists, they only use MEA. It grew very slowly on MEA, almost don't develop the reproductive structure. It's very hard. We have to use the VA to speed up. The morphology is the same method. And this one, we use the 4 loci and to determine the, whether it's new. But for right now, if you want to describe a new species, the morphology and the molecular part have to combine together. Although there's one journal still, you can publish purely on morphology part. Then they are the same, but then we have to use mega 7. Right now, mega E is 11. 11 is actually is much better. Mega 10 doesn't work. Usually I don't use, I still use Mega 7. Bayesian inference, then also I mentioned you use 2 different methods to confirm whether it's something new or not. Then it's very, very important for this. And this is the tree. I have to walk over, otherwise I cannot see. Okay. This is the new species. Okay, if you can see, it's very close related to the type species of this genus. Then the rest, we have to, include a lot of close related species to make sure they have a strong support to separate them up. And this is the Chaetomium globosum. This one actually is very common. We can find it in indoor environment. Wherever you have water damage in indoor environment, then there are several fungi you can find. Long-term water damage, guarantee you will find the Striatibotrys, then Chaetomium globosum, and Memnoniella. And there are several fungi is very, very common in indoor environment with water damage. Then from here, we can see this is the new species and separated with the one include here 4 athletes. And this is where where the sample was collected. But it took several years to work on to figure out. You can see they are collected in 2012. And the morphology. Then from the description of the name of the genus, we know should have a theta, but this one doesn't have it. Okay. Another is the aerial CFA is very different. It's very, very rough. Eventually figured out it's not solid one. It contains some kind of a liquid in it. Can, open up, then release that. And this is why the function we really don't know for this one. And Canadian form, there are some differences, and I will show you later on. Then the mother cell, we can see those in bold show the differences. And Canadian is very similar, and shape and the size and thick wall, and no sexual state developed. Okay, but when we talk about fungi, always have to talk about, the asexual stage, sexual stage. Sometimes we can find both. Most of the time, we find only one. And this is the one, okay? Can you see on MEA? It's tiny, doesn't develop. VA, you can see. Then it's a huge difference. This is why we have to, use a different method. Then this is the type species of this. It's close related. This one have a CD, but this one doesn't have. People think, oh, this look like a CD, very dark, rigid. Then, but it's not. Why we don't call it a CD? Because on those hyphae, those structures develop. On the CD, then they don't develop any reproductive structure. Okay, and that's the part. This is a Canadian. You see the blister-like stuff. Then here on this is the one open up. And based on those, or another characteristic is this, the mother cell. Can you see here? What is the difference? It's quite different. The size, the shape is very different. And this is why, we publish as new species. Okay, doesn't work. I have to use this. Okay, now the question is, what should we call it? Because the new nomenclature creates some kind of a chaotic situation. We have to choose when you have a sexual, asexual, which one publish first, and whether we should preserve the name or conserve it Then, but this one, you know, we know the sexual stage related to this. This is published much earlier, and this is later. I noticed there's one new paper published. I cannot get. Then I can find the sequence data, but I want to know because they move some sexual stage to this. I want to know why, what kind of reason make them to decide they should want to use this name, not the one with the priority. It's very, very interesting. Okay, next one is a very interesting one, Fusarium. Then Fusarium, this one, not like a previous two, only saprophyte. This is a severe plant disease. And before we talk about, you know, Fusarium, then the information is very, very interesting. Fusarium, you know, then described, you know, you can see more than 200 years ago. And right now, then valid species probably 400 and 23 species complex. Then, but if you check the index Fungarum, you will find 1,700 names published in this genus. It's huge. But the Dutch group, well, this one, you know, is very, very important from economy, health, not only human being, animal also, and plant pathology. Then many plant diseases caused by Fusarium. We can tell you a whole bunch. He worked on Fusarium for many, many years. And the Petrocruz, then that group published a paper, then divided the Fusarium into many, many different genera. You can see, but Caryandana and some other mycologists have strongly different opinion. Then this why is controversial. And but this in this study, we struggle which name we should call. Should we follow this group, or we should eventually, it's a plant disease, it's easier for plant pathologists to use it. We decide to stick to Fusarium. Okay, this is the story about this. This disease actually is from the city. I did my undergraduate study there, and during the field trip, one faculty member lead two undergraduate student to field trip, then found the disease. Oh, I jumped ahead. The Fusarium allosity can produce mycotoxin, and you know, for human disease, you know, if you remember a few years ago, then the people wear the contact lens, then contact lens solution, then contaminated by Fusarium cause some infection and become big news. But artificial protein, then extract produced by Fusarium develop into some kind of this kind of a product. It's available in North America, but I have to say I never tried. I never. Well, yeah. Oh, you tried? Yeah, I never died to try. I have to say, you know. But my concern, you know, then if contaminated, you cannot tell them apart. And then the one eventually for the one we isolated is belong to this species complex. It belong to Solana. And this one contains probably over 60 different species. And but we can see this species complex can infect many, many different plant hosts, close to 600. Probably right now, I would think will be over 600. I know we have some, you know, to publish, probably will be over 600, you know. Oh, that one is a different clade. Okay, I have to withdraw that statement. But you know, 2020 they found this. Then we want to do all this for objective, you know, and characterize, identify the pathogen, determine, you know, whether it's a causal pathogen or not. Then actually this part is very interesting. Want to determine whether it's cause systemic infection or not. Then this one is okay. For the people, from China, maybe know this Pukou. Pukou is the district across Yang River from the major city. And they established a new park. This is why they transplant some roses there. And during the field trip, I mentioned they found many roses died. Then the faculty member collect some sample, bring back, and talk to the plant pathologists in the department. And this is why the research started. Then 4 athletes were isolated. But there are additional athletes, but it didn't characterize, do the thorough work. We cannot count those. And we sequence, 3 loci and to do the analysis. Then when we submit the paper, they require we have to do this analysis to confirm and it's a new species, and which we did. And we do the pathogenicity test in 2 different methods to confirm it's truly a pathogen. Here is the one, the symptom in field, you can see killed the whole plant. Then in short period of time. Then you can see when you put it into a moist chamber, the mycelium can come out from everywhere, the cross section. Then this why, make us to think, is this a systemic infection or not? Okay, then for the method for the systemic characterization, there are several steps to follow. Then to use the green fluorescence protein to label the fungus. Then to indicate, we can take a look, collect samples, then to show where they reach when the root is roots are infected or inoculated. Then okay, this is the tree. We can see on the top, this one really separated from the close related. Later on, I will show you the morphology, how to separate them up. Then, and this is the result. And there's another one, include Wimo taxa. Then, but there's no space to show it. Sometimes works, sometimes don't. Okay, this is the what we isolate, then describe, then you can see this is a tab, then and this is the faculty member, then he's the one of the author, and this one is very interesting. They developed the sporodokia. Then this is why I bought this. This is the key characteristic. And on this, the clinician on the area heavy and emerge heavy, then they are quite different. Okay, another characteristic is polyphyletic. Then this one, then that means one phylet they can develop, from at least two different loci to develop conidia. Then other, is you measure the shape, you describe the site, then and macroconidia. Okay. For Fusarium, then people consider, they can develop quite a few different conidia, macroconidia, macroconidia. Some people even use mesoconidia, and plus chlamydospores. Then those are all useful to separate, to differentiate different species. Then from morphology. Some develop the full cell, some don't. Then this one, although I think it's quite obvious, but one of the reviews think is different. We have to follow that, you know. But they do develop the chlamydospore also. Okay, those are macroconidia. This is macro, and this is chlamydospore. And when they develop the sporodokia, you see the cuneiform is quite different compared. Oh, this one from the aerial hyphae, although this is in bundle like a cord. This is another characteristic to tell them apart. Okay. Okay, close related. Can you tell the difference? This one, macroconidia, no food cell. Oh, I should point out which one is food cell. The not of this one. Okay. And we can tell the difference. And this part is different also. And clearly we can separate them up. This is the Fusarium. Okay, if we follow the Dutch group, then we have to place this into this genus. And but we don't know in future, you know, if we can settle down the difference, then that's a different story. Okay, the pathogenicity test, then 3 close related hosts, then they can be all infected. Actually, the cherry tree, you know, then infection is stronger. The stem, you can see this one only do 1 athlete, then 3 different photos to show this is control, this is the one, and on the surface, cut it open to see the inside. The root, the symptom on the root, and they can infect all the part of the plant. Okay. This is the one to show that if we successfully transform the fungus with the protein, then under fluorescence microscope, you show this color. This is on DIC, combine 2 together, show this one to prove it. Okay, this is the one to show the systemic infection. The root was deep into the Canadian suspension, then planted. After that, this is the control. This is the infected one. This one close to dead. Then this is on the leaf, on the stem. You can see when collect all this, re-isolate the fungus to show all have show the original the pathogen we inoculated, then to show they really cause systemic infection. Okay, next one. This is actually by rich coal, then cause a big problem. But this one is not a true fungus. Okay, although I use this, some people may call different content. Different mycology we may prefer different names. Then right now about this number described species, but the one paper estimate then probably about 500 different species in this genus. But over 80, then they cause severe problem. If you think about, you know, Irish famine, plant pathology, and this person, then because Irish famine is caused by Phytophthora infestans, then and in 1840s to 1845, then from there, and DeWei is the one people consider is the founding father of plant pathology because that single disease, then big epidemic, then caused a loss problem in Europe, in Ireland. But we can see there are 16 species actually from freezer fir. Freezer fir is one of the major species of Christmas tree production in Connecticut. if people know Boston quite well, know this monument or statue is the gift from the Irish Ireland, then in 1990s, if I remember, then and show the big family and many families are separated. Then and actually in Boston there are 3 different statues, you know, in memory of this big family. Then because the Irish population in Boston is very significant, then probably a lot of people, I don't know how many people here, then have Irish background. During that pandemic, you know, the Irish population dropped to half. People either died or moved to other countries, especially in United States. For this one is a very interesting one. More than 10 years ago, Rich planted a whole bunch of Christmas trees. Then some are freezer fir, but later on, then every single one died. Then he tried to figure out what's the problem, then and take some time to able to purify the acid, purify. We try many different medium, it doesn't work. Then, but eventually find a way, a very old way to acid. Then, but for molecular part, then that's the loci we sequence, and we did the pathogenicity test to prove it's truly the pathogen. Okay, then okay, this is the field. You see every single one, all dead. Then even right now, you go to Christmas tree farm, even my plot in Windsor, the mushroom cultivation plot, because I want to, you know, develop the intercropping system. The Christmas tree, quite a few dead. Then I know it's caused by Phytophthora. Whether it's this species, I don't know. Okay. Then the methodology is very interesting. You see, what we have to use apple. We try quite a few different medium, doesn't work. Actually, I got frustrated one time. You know, I went to Harvard to use the library. I talked to Don Pfister. He said, "Why don't you try the oldest, very old method? Use the apple." Then we did. Works. We isolated. Then that's the way. You see the method, old method published 90 years ago, 91 years ago, but works, still works. Then, and right now, Rich continue work on this. He actually some stuff, the Phytophthora, some still have to use this to purify, to get the pathogen. Then, then after we got that, we can use VA and or PDA and to work with, or have to speak it up. Holy smoke. Morphology, well, similar methodology. I have to skip this. Okay, this is the apple we use. Usually we use this, insert into this whenever, you know, then developed, then we will re-isolate, then transfer into VA or MEA to purify it. Okay, this is the symptom. Okay, usually the branch drop and bend down, then show the symptom. When you cut it open, you can see the infection. Over here also, you can see. Okay. Then when we do this, then actually we cut this, you know, open up, then insert the inoculum, and over here also, that's the inoculation we did. Okay, this is the tree. We see this is the clearly separated with close related this one, and it's quite obvious it's a new species. Then right now this species was found in Pennsylvania in the forest. Then they isolated from quite a few different tree species down there. The paper I think was published. Then, and well, that means It's not introduced, probably it's indigenous already here. Morphology is actually from the Connecticut, and we do got the permission from the farmers whether he want to his farm name show up here. It all doesn't matter, and go ahead, but we have to get permission. Morphology, actually 2 characteristics. Then the oospore is a sexual spore, okay? Then the wall is very, very thick and it's very interesting. And the brownish yellow, this is another characteristic. The male part is this, doesn't show much of characteristic. And asexual stage, we are able to get this, and those spores supposed to swim in spore, but we cannot find the flagella. It's very interesting. That's something. There are some, and the chlamydospore also we found this. This is the measurement. Okay. 2 characteristics. Oospore in the oogonia, the color is golden yellow. Well, actually, this one is not a very good one. I have some photo. It's really show the golden color and a very thick wall. Can you see the wall here? Oh my hand, I even use both hands still not steady. Oh, yeah. Wow. Okay, this is sporangia, immature one. This is empty one, and inside of the empty zoospore, they can develop another one. Okay, and this is a zoospore, and I cannot find the flagella. They're supposed to swim in spore, but and over here zoospore. But mycelium could be very interesting. It's very complex. They form this kind of a strange tab. I don't know the function of this. This is for future. Mycology is a very interesting field. I do like it, but there's a lost hurdle in it. And right now, the delineation of a species concept, even the genus level, how to define. Then people tend to split up. Some people tend to merge together. We have a lost lopper, splitter. Then doesn't matter you use a molecular method or morphology, then always people have a different opinion. But evolution play an important part. That's why right now we always find some new species or new pathogen. Then one story is, I do have a little bit time. Then we found the Chinese fir and plan to get alternate with the rice. Then the rice pathogen eventually jump onto Chinese fir, become pathogen. Before it's harmless endophyte. The last couple of years, they found cause some severe problem. That's part of evolution. When one season you plant the Chinese fir, another season you plant rice, then force them, to evolve. Then at the same time, then this part is very important. When I do the Striatibotrys study, I borrowed a lost herbarium specimen, then really open my eyes. You can compare, but you will find some problem. Then one time I borrowed one is type specimen from New York Botanic Garden. On the same one, I found 2 different species of Striatibotrys. I scratched my head. Which one he described? I have to make a call, It's very interesting. But another thing is gene bank. We know gene bank, there are some problem, and about 20% to 28%, depending on which author you think is reliable, then they are wrong. But when people use the blast method to try to identify, use one locus, we could got it wrong. I remember one time I checked one fungus, eventually isolated from bamboo. Then the sequence ITS is bamboo, not a fungus. But the 2 part, they won't allow third-party annotation. If there's any problem, you cannot correct it. You just have to, you know, keep to yourself. Evolution and the new emerged diseases. Then there are many new diseases. You check the disease notes in plant disease. Every issue, there's a lot. Okay, that's the problem we are facing. Then I think that's okay. My collaborators and more than this, but they relate to the 4 species I described here. This is the one I collaborated with. Then this is a slime mold, but it's an indoor fungus. You can find when you have water damage in your house, you may find this one. Okay, any questions? That's the last one. Yeah.
So there seems over the past certainly 20 years as this tipping point went from morphology-based speciation to more
right.
And so where do you think it's going to end up 10 years?
Very good question. I don't know. Unless the whole genome information all available, we may be able to tell. The thing is right now, molecular data is not bulletproof. Then some people use the molecular method, then found the axial stage, asexual stage linked together, but later on prove it's not correct. If you only use 1 or 2 or 5, you still end up maybe not that correct. I don't know. Yeah.
So kind of related to that, your housekeeping genes that are helping catalog all of these indoor fungi, could that be used with like a larger data sets that are available to figure out if these new fungi are widespread, widely distributed, or
yes,
or is it too complicated genetically?
I should have repeated your question. I forgot to repeat your question. Yeah, the question is, you know, then the housekeeping gene, whether we can apply to a broader sense to determine whether they are, well, in terms of distribution and the other aspect. Then yes, my answer is yes, it's possible, yes. Then when you use the gene bank, then if you check the data, a lot of information there, where they collected and the host or substrate, then you really can put out a lot of information there. Yes.
So if you're looking at morphological differences and phylogenetic differences, how do you know when you have an appropriate sample size to actually make the declaration that it's a species?
Proper size. Okay. The question is when you use the morphology and the phylogenetic method, then what is the proper sample size? Okay. Right now, the plant disease before only need 1 athlete. Right now, they require 3. If you want to describe a new disease or new species, but it depends on the editor. Some editor will let you pass with only 1, and some require 3. Probably you can answer. You are the senior editor. I don't know the policy. Then, because based on my experience, when I submit the manuscript to the plant disease or, you know, phytopathology and the European journals, then few years ago, for sure, 5 years ago, you can use 1 single athlete. But right now, they usually require you have to have multiple. But go to extreme, we submit 1 manuscript, we include more than 60. Okay, but they require you have to do all the pathogenicity tests on every single athlete. Holy smoke! I know, you know, for graduate students, whether they have the time to do so many or not. But, you know, review, they require this. This is why tonight I have to have online meeting with the group, you know, and to discuss how to handle this.
Just to top of that question, isolates them off to describe specific fungi or already.
I cannot hear you. Okay.
Your own opinion.
Yeah.
Isolate. Is that enough to describe
Okay, depending on what kind of fungi you work with. For macrofungi, mushroom and wood fungi, you have to have multiple samples. From the history aspect, the hypomycy doesn't require that. When you have culture, you have many reproductive structure on the plate. You can see the variation. Then if they show, some people may disagree, but that's the tradition. They may change, but sometimes you only got one. Sometimes you don't even have one. You are not able to isolate. I have a few species published only based on the morphology. I cannot isolate. It's dead. Then another question is, in future, if the technology, I know the technology is available, but right now it's very expensive. Single spore sequence. Then I know one study related to Irish famine. They did, single spore isolate to study the historical specimen from the different herbaria. Then it's a very good one, but it costs a lot of money. They got a huge grant to work on it. I don't have that one kind of grant to work on that. I have to see, Yeah.
You feel the changes we're making to managing indoor spaces in buildings because of COVID are going to affect the indoor problem?
Yes.
In what way?
Okay, very small particle. Then if you follow that, some people, CRHs, industrial hygienists, they prefer much better filter than HEPA filter. The HEPA filter will cost a lot of energy already. But if you want to have the filter to screen out the virus, then cost a lot of energy. At the same time, then will change the CO2 level and humidity level, the whole bunch. The building design have to, change along with it. Any more questions? Okay, thank you.